Good morning, students. Today we are going to perform a science to practical, and our aim is to study of lactobacilli present in curd. In common language, the curd is known as dahi. So in theory lectures, we had studied about different types of microorganisms, useful microorganisms and harmful microorganisms. Useful microorganisms means what? Those microorganisms, they help in our daily life. From that useful microorganism, the one is that lactobacilli, and the lactobacilli in which lactobacilli is present in curd. So we have to see that lactobacilli by performing the practical. So how we will perform the practical? First of all, I will explain you in theory, and afterwards we will perform that practical. Okay. What we have to do? We have to take a slide, glass slide, and on that glass slide we have to pour a 1 drop of curd. Okay. After that pouring a drop of curd, we have to pour a methylene blue drop. After that, we have to remove extra curd and extra methylene blue. What we have to do? We have to remove the extra material that present on the slide, and we have to put cover slip on that smear. So what is this? We had made a smear, and that smear we have to see under the compound microscope. So what we will see? We will see lactobacilli present in curd. And how these lactobacilli we can see? These lactobacilli are rod shaped structure and blue in color, because it takes the color of methylene blue, and methylene blue is in violet color. So methylene blue, due to the methylene blue, the lactobacilli are of purple color or blue color. Okay, so how we will perform this practical? Let us see with the help of practical. As we know that our aim is to study the lactobacilli present in curd. So which are the things we need in a practical? We need a curd, we need a methylene blue, we need a slide. This is glass slide, we need glass slide, and we need cover slip. Okay, and dropper also. This is a dissection box. In dissection box, all things were ready. From this dissection box, we have to take only dropper. Let us perform the practical. Okay. So, first of all, what we have to do? We have to take a glass slide and put drop of curd on this glass slide with the help of dropper. Okay. We had taken this drop of curd. What we have to do? We have to put this drop of, first of all, what we have to do? We have to take a drop of curd and we have to put on the slide. After this putting a drop on a slide, we have to put a drop of methylene blue. Okay. After this putting the methylene blue, we have to wait for 2 minutes. Okay. After 2 minutes, what we are going to do? We have to remove the extra material, that is curd and methylene blue, and we have to wash this slide, but we have to wash this slide properly, otherwise our smear will damage. And when we are making these things, this thing is known as smear. जो हमने ड्रॉप डाला है ना दही का, और उसके ऊपर हमने मिथिलिन ब्लू जो डाला है, अब हमें एक्स्ट्रा जो है उसे रिमूव करना है. जो एक्स्ट्रा हम रिमूव करते हैं, एंड द थिन लेयर दैट वी कैन सी, दैट थिन लेयर इज कॉल्ड smear. Okay. Let us watch this slide. We have to dry this slide with the help of blotting paper, so that our smear will become thin and we can observe that lactobacilli properly. Okay. Now what we have to do? We have to put this cover slip. This is a cover slip, and we have to put this cover slip on this smear. Okay. How we have to put this cover slip on the smear? Properly we have to put this cover slip so that air bubbles can't enter in that smear. Now this is a temporary slide. Okay. Now this slide we have to see in a compound microscope. Let us see this slide. Come fast. Students, you know now this is a compound microscope. Come here, girls. Now we are going to focus this slide under a compound microscope in 10x. Okay. Yes, you can see this rod-shaped bacteria that are lactobacilli. One by one come and see this slide. Okay. From this practical, what we have discussed today is we see that lactobacilli that are present in curd. So today's practical is over. Next practical we will take in next lecture. Okay? Yes,